has been influenced. Okay. So they have been, they will be influenced by our daily activities, for example, nutrition uptakes or medications. And the activity of these organisms also will influence our health as well. In some extreme conditions, for example, the allogeneic hematopoietic cell transplantation, or we will say bone marrow transplantation. So this is a procedure that is a cure therapy to cure all those, a lot of those hematopoietic malignancies, such as leukemia or lymphomas. And the recipient of these in these procedures will receive very high dose of chemos or total body radiations, and they're on highly immunocompromised conditions. And these conditions can damage the barrier integrity and lead to the microbial dysbiosis as well. So these patients are highly prone to all sorts of infections, and they are required to be on different kind of prophylactic drugs, high dose of antimicrobial drugs. So in the hospital where I did my postdoc, Memorial Sloan Kettering Cancer Center, they have a very special procedure for how to protect these patients. So this is an antimicrobial prophylaxis profiles of they have. So this patient will start to have the antifungal prophylaxis or antibiotic prophylaxis right together with the conditioning like radiations or chemos when the patient start to get become immunocompromised. And the immunocompromised situation will last to another week or two, at least a week or two after the donor cell infusion, because it will take at least a week to 2 weeks for the donor cell to infuse and expand to reconstitute the immune system of these patients. And so during the whole neutropenia time, this patient will require to be on antibiotics and antifungals. And in Memorial Sloan Kettering, normally the patients under this procedure will be put on antibiotics, for example, IV vancomycin or fluoroquinolones. And for antifungals, they have the special the combinations. At the beginning, they will start with microfungin, that is a kind of candidin drugs target the fungal cell wall synthesis. And then they will switch to azole drugs, for example, voriconazole, posaconazole, or salbuconazoles. The reason they do that is because this patient are not only on these antimicrobial drugs, they are also, some of them are on these conditional chemo drugs. And these chemo drugs could have drug-drug interactions, and azoles has more drug interactions with these chemo drugs. So these oncologists, they were worried about that this drug interaction may affect the chemo drugs. That's why they use more safe drugs, such as microfungin at the initial stage when there are still high dose of the chemo drugs at this point. And as the chemo drugs metabolite out, flushed out, and they will switch to azoles because these are azole drugs not only can protect the patient from Candida infections, but also can protect the patient from mold infections such as Aspergillus fumigatus. In some conditions, this patient will under very long neutropenia times, even longer than a month. And they may have like very complicated situations such as liver situations or renal situations. That's why some of these patients will be on microfungins for even longer than a month, and they will be put on all different kind of antibiotics like peptides or carbapenem, these very strong antibiotics. And all these antibiotics, antifungal drugs will shape the structure of human microbiome together of the bacteria and fungi together. And in terms of fungi, I forgot to mention that for microfungin, this patient received microfungin through intravenous route. And we were curious, like, what is the microfungin level in the feces? Because these are the in the gut, because the intestinal tract are adventitious for a lot of different kind of microbes there. And that's why we picked, we tested the fecal level of microfungi from over 100 samples from 13 patients. And here, each dot represents the level of microfungi, microgram per ml level of each fecal samples, and they were plotted in terms of the transplant day. So the patient will start on microfungi sometimes a little bit earlier than 7 days than the chemos, and it will last usually till 7 to 10 days after transplantation for the microfungi. So we can see that during the microfungi prophylaxis, the fecal sample level of microfungi is relatively high. So how high is it? I'm going to give you a chart of the micamine. This is a commercial chart of the microfungi, the drug's commercial manual. And you can see that most of these fecal samples during microfungi prophylaxis, the microfungi concentration is higher than 1 microgram per ml. And this would covers quite several common fungal Candida species, such as albicans, tropicalis, glabrata. And there's one species stands out, that is a parapsilosis. So we can see that the parapsilosis species is in general more tolerant to echinocandin drugs compared to other Candida species. And this reflected in our microbiome studies on these patients. So together with my colleague Thierry Rowling, we did a cohort study of over 150 patients with together of 1,200 fecal samples that collected from 10 days before and to 30 days after transplantation. So these are the ITS sequencing result of all these over 1,000 fecal samples. And we can see that there are 3 clusters of samples that by sequencing has a high, has a kind of like one fungal species domination, like dominated by Candida albicans, this pink one, and dominated by the Candida parapsilosis, and dominated by Saccharomyces cerevisiae. Meanwhile, we also cultured each samples on the agar plates with antibiotics, so only fungal CFUs can grow out. And we can see that for these 3 clusters of samples, only the cluster of samples with Candida parapsilosis dominations coordinated with a high frequency of culture positivities. So this suggests that the domination or expansion of Candida parapsilosis is real. We can validate it not only by sequencing but also by culture, and that in this patient, this parapsilosis colonization or expansion is very frequent, likely due to the microfungi prophylaxis selection. And when did this when did this parapsilosis domination happen? So we compiled all of the over 1,000 fecal samples on the timeline of the transplantation. So we can see that these are suggested, the colored bar suggests that the samples that with Candida parapsilosis domination and or Candida parapsilosis or Candida albicans domination. So we can see that the parapsilosis domination happens mostly during the 2 weeks after the transplantation. That is the neutropenia time as of these patients. And what will happen to these patients? We can see in a survival study of these patients, we can see that. So for patients with parapsilosis domination in their intestines during neutropenia time after transplantation, they will have a much worse overall survival compared to those without parapsilosis domination. And in total, there are 12% of these patients had this intestinal domination with parapsilosis during neutropenia before engraftment of the donor cells. And 3 of them developed parapsilosis bloodstream infection. So what will these patients look like in their gut microbiome? In an earlier study from our group, excuse me, we retrospectively pulled out a patient with candidemia during their neutropenia time after transplantation, and we examined the microbiome of these patients. So the gray arrow and the dash line represent the transplant day, and the black arrow and black dash line represent the day of blood culture tested positive for Candida species. And we can see that, which means the detection of bloodstream infection of Candida. And we can see that almost like a week before the detection of blood culture positivity, there is like a single fungal species domination in these patients. So, which means that if we can monitor this patient more closely, and we can predict which patients are more at risk or are more likely to get infections. And very interesting is that we, in parallel, we also analyzed the bacterial communities of these samples, and we found that when the samples have high fungal components, high Candida abundance, these samples usually have a significantly lower bacterial density and bacterial diversity. So the intestinal Candida domination or Candida parapsilosis domination in these patient cohort has been linked to the loss of anaerobic bacteria in the gut of this patient. So with these two studies, we basically build up a connection that if there is a fungal infection, bloodstream infection in this patient, we can almost be sure that this patient had a fungal intestinal colonization before their bloodstream infection. How about the other way? So I just showed that there are 12%, 19 patients had fungal colonization, but only 3 of them developed bloodstream infection. Why is that? I'm going to show you an example, a patient example. This is my favorite patient. So this patient has been on microfungin started before transplantation and lasted at least for another 2 weeks after transplantation. And this patient had a bloodstream infection detection of Candida parapsilosis at day 14 after transplantation. And even as early as day 2, this patient started to have the fecal isolates of Candida parapsilosis. So this patient started to have Candida parapsilosis gut colonization at very early time. When we analyzed the whole genome sequence of the fecal isolates and the blood isolates of this patient, we found there are 3 clusters of these patients, and they are I use the color yellow, blue, and orange. And we can see that the bloodstream infection isolates belong to the orange cluster, and the fecal isolates has been shifted from the yellow, blue, and to the orange. So what's the difference between these isolates? I would like to mention here that none of these fecal isolates or blood isolates from this transplantation or from this patient or the other patient has been tested as resistant to microfungi. So they are all susceptible by microdilution or e-test. But if we use a higher resolution assays, that is the population analysis profiling, the PAP assay. This PAP assay, what we did is that we played 1 million fungal cells on agars that containing different concentration of microfungi. So this is a concentration of microfungi. These are not the time or whatever. And we measure the CFUs after 3 to 4 days, because this resistant if there's any resistant colonies, it will grow slower. So we measure that after 3 to 4 days, and we divide we calculated the survival CFUs and divided by 1 million, the initial inoculum, and it will calculate the survival rate of this organism. And we can see that for the orange and blue isolates, by e-test, there's basically no difference of their minimum MICs, minimum inhibition concentrations. But by PAP, the population analysis profiling, we can see that for the blue, for the susceptible, the blue isolate, the true susceptible isolates, at the concentration of 2 microgram per ml microfungin, all of the cells, 1 million cells, will be killed. There's no survival CFUs. But for the orange ones that we call the heteroresistant ones, even as high as 32 microgram per ml, there will still be a very low ratio of the cells, about 0.01 to 0.1% of the cells, that can still survive and thrive, that because they can grow out colonies, so they can divide, they can proliferate under the high microfungin pressures. So this is a very interesting phenotypes, and we think this could related to the outcomes of the infections that because the infection developed during microfungin prophylaxis. Therefore, I look back to all the patients that I have studied for their gut microbiome, and that I have their sequencing data, their culture data of the gut fungi. And in total, I can identify 25 patients that had intestinal parapsilosis domination or in colonization by culture validation before engraftment. And among these 29 patients, 15 of them, the colonized organism has been tested by PAP and been defined as they are all susceptible cells, susceptible isolates. And none of these patients that are colonized by susceptible isolates eventually developed a bloodstream infection. And for those other 14 patients that are colonized by heteroresistant isolates or a mixture of heteroresistant and susceptible isolates, 5 of them developed bloodstream infection. So this intestinal colonization of heteroresistant isolates really is the true risk factor for microfungin breakthrough infection in these patients. And when we put all these data and together with some other supporting data together and submitted to Nature Medicine 2 years ago, the editor, sorry, I should introduce like the hypothesis. So the hypothesis here is that it's both orange, the heteroresistant isolates, and the blue isolate, the blue cells, the susceptible isolates, they can both colonize in the gut, and they have equal chance to transmit from the intestinal to the blood vessel. And because microfungin has much higher concentration in the blood stream, so the high concentration of microfungin can kill true susceptible cells. And in contrast, the heteroresistant cells, most of them strains, they can also be killed. But a small portion of that can amplify even in the presence of microfungin. Therefore, this is how they develop the bloodstream infection. And yeah, as I just described, when we initially submitted this story to Nature Medicine, they said, okay, yeah, it's very interesting. So how about the generalization of your observation? Is this phenotype that particularly observed in Memorial Sloan Kettering Cancer Center? Does it exist in other isolates? So we reached out to our friends, Geraldine, our long-term collaborator, Geraldine Butler, a very supportive physician scientist, David Endis in Wisconsin Medicine. And I also established new collaborations after I moved back to China. So I reached out to a physician scientist in Beijing in the Peking Union Medical Research Center, and they have a large collection of clinical fungal isolates there. So in total, we collected over 200 Candida parapsilosis isolates from Asia, from Europe, from North America, and we tested their phenotypes by PAP, and we also sequenced their whole genomes. And this is a very nice circle with a lot of numbers. What a sentence for short is that we can find heteroresistant isolates in European isolates. We found heteroresistant isolates in China isolates, and also, of course, in North American isolates. So this is a phenotype that exists not only in the Memorial Sloan Kettering Hospital, but also in hospitals in Europe, in China, and probably in a lot of other countries in the world. So this is not a unique phenotype, but this is a more generalized threat so if there's a hospital, for example, in China, they use microfungal or some other kind of drugs for prophylaxis for say 10 years or 2 decades, they probably will see more parapsilosis breakthrough infections as well. Okay, so about this interesting phenotype in parapsilosis, what do we know about that? We know very little. So through our whole genome sequencing, we did not detect any mutations in the known resistant, echinocandin resistant related genes, such as the drug target FKS1 or the MSH2 gene that respond for the mismatch repair that has been reported by David Pauline's group that may play a role in the Candida glabrata heteroresistance or tolerance to echinocandin drugs, but there's no detectable mutation in this gene as well. And there's also no proper clinical diagnosis approach for us to detect that in the clinic, because the PAP assay is the standard, it's gold standard way to detect heteroresistance in the lab, but it will take much longer time. The patient will not be able to wait for 3 to 4 days. The clinician cannot wait for that to that long time to choose the correct antifungal drug to treat the patient. So how can we handle this issue? I was really lucky that I can find a very talented collaborator when I was in MSK, Dr. Chen Liao, who is going to be a assistant professor in Dartmouth College next year. And he's a assistant biologist that is very good at modeling biological questions into and dissect that with his mathematical tools. And I was also very lucky to recruit Yuan Tang, who is a postdoc in my lab now. She also is very enthusiastic in the Candida parapsilosis genomics. So what we has been doing in the past 2 years is we're trying to map the genomic features that are highly correlated with the heteroresistant phenotype. We hope this effort could offer us some more insight about the mechanism and the maybe can direct us to find a better diagnostic approach based on these genomic features. When we talk about genomic features, there are different levels. There are SNPs, there are copy number variations at ORF levels, or there are aneuploidies. In terms of when we're trying to look for the genomic features and when we're trying to enumerate the copy number variations, we actually came across some very challenging parts because, for example, the current way or the previous way to calculate copy number variation is that we calculate the average coverage of an ORF and we divide the average coverage of the genome and times 2 because Candida parapsilosis are diploid. And take this ORF as example. So this is an ORF on the chromosome 6 of parapsilosis, and this is one isolate that this ORF it has a truncated version. So the half of this ORF is deleted according to the BAM files figures. So with this previous way to calculate CNV, the CNV of this ORF is 1. Now, does this CNV equals to 1 means like just only one allele and there's a 1 now mutation, or there's like a half truncation like this? So this is not very clear. And we were really bugged by this previous approach to calculate CNV. And Chen developed a two-factor method by numerate not only the base pairs, but also base pairs of each the coverage of each base pair, but also trying to calculate whether it's a full but also trying to describe what is the intact situation of this ORF. So basically what he did is that he calculated each base pair's coverage and make a plot a distribution, and he applied a Gaussian kernel process and trying to find how many peaks are there. And by calculating the peaks and the number of the coverage at the base pair, the number of the coverage of the copy numbers, and he could plot the copy number variation as 5 different types, as normal, partial amplification, full amplification, full deletion, or partial deletion. So we calculated the copy number variation, the CNV parameter, in a more defined way compared to the previous way that just give you a number of an norm. Why we take care of this, why we take so much effort to dissect the CNV, it's because that when we analyze this, we can find that there are some features that are highly associated with heteroresistance, and this is a CNV actually. So this CNV is very complicated. Also, it has both all this different format. It has normal, has full amplification, has partial deletion, has full deletion. And we can see that only the full deletion is highly correlates with the phenotype of heteroresistance in these clinical isolates. And with this, Chen was able to develop a proof of concept machine learning based approach. So what we did is that we divide our isolates, and first though, we clean up with the isolates, our isolates, by remove the redundant isolates by comparing the similarity of their genomes. So we clean up all the redundant isolates, and there's only 160 isolates left. And then we put part of them as the training data sets and part of them at the test data sets. And we want to run the machine learning approach with a limited number of genomic features and see whether we can use that to predict the heteroresistance. So our final score is that we can reach about 75% of F1 scores, which means that 3 times out of 3 out of 4 times for the prediction, we can get a correct, either it's heteroresistant or it's true susceptible phenotypes. So it is not perfect, but it's still better than nothing because the current approach to detect heteroresistance is 0. The successful rate is 0. So we hope that with a more diversified clinical isolates to be tested, we can find more accurate genomic features that associated with this phenotype and help to develop the better clinical diagnostic approaches. So the summary of this part is that first, we know that from this microbiome study, we can find that the intestinal colonized fungi is the major reservoir of the fungal bloodstream infection in these particular patients, in the allo-HCT patients. And the mycofungin heteroresistant fungi is the true driver of breakthrough infections. And we also try to optimize the genome analysis approaches and try to identify the genotypes that are highly associated with these phenotype, and hopefully we can have more idea about the underlying mechanisms and help to develop the diagnose approach. So that's the study I've been doing in the past 10 years. It's actually exactly 10 years since I studied this project in Tobias lab. And I've been in China, back to China for more than 3 years. So what about my own research group? Because that's the resource in that hospital, and I need to have my own stuff here back in China. So I go back to the fact. So this is the facts. What are the fungi and where are they on our body? And what are the importance, clinical significance of these colonized fungi? So we studied intestines, and there's another part that's very important, that is the vagina. So vulvar vagina candidiasis is one of the most frequent human fungal infections, and the VVC is caused by Candida colonization in the vagina. So the Candida colonization, some of the Candida colonization could cause itchy or burnings in these kind of symptoms. And there's a high incidence of recurrence of this infection. And what's really interesting to me is that there's only about, at one cross-section time point, there's only about 15 to 20% of women that had detectable Candida vagina colonization at this certain time. So like in the audience, all the ladies, not all of you have the Candida in your vaginal tract. There's only a small portion of us will have it. So what is this what is the unique of these individuals that has Candida vagina colonization? I would like to, by studying that, I would like to have a small introduction about what is a vagina ecosystem. So this is a gram staining of the vagina swabs, and we can see this is the epithelial cells, and these are the bacteria. So the most common bacteria in the vagina ecosystems are the lactobacillus. So they produce a lot of acids. That's why for a healthy vagina ecosystem, the pH is usually quite acidic. It's usually between 3.8 to 4.5. And this low pH can inhibit the bacterial passage and prevent the bacterial infections. And the previous studies on the vagina microbiome divided the vagina ecosystem, vagina bacterial community state type by 5 state type. So 4 of them, CST1, 2, 3, and 5, are dominated by different lactobacillus species, and CST4 is dominated by other like Streptococcus or Gardnerella, those kind of more pathogenic bacterial species. These are the bacteria. Okay, so here's a slide with fungi. This is an albicans hyphae there. So by seeing this, by just looking at the slides, you probably cannot tell too much difference about the community because the resolution is not that high. And what are the fungal community state types? So we know there are not that many, not high ratio of individuals that has colonization, but there could be different Candida species there, right? And what are these? And when we look at literatures, we found that there's really very minimum amount of high quality data that can show that. So in terms of answering this question, so by collaborating with a local hospital in Shenzhen, and we were really, my collaborator, Dr. Li Juanwu, and we were really lucky to recruit a very talented postdoc, Yanni Fan. And we built up a cohort with pregnant women that we can build up a longitudinal vagina sampling throughout their pregnancy. And this is more like a case control cohort because we did not enroll in a participant just evenly. We actually select because we want to check more of the fungal dynamics there. So we select a more fungal culture positive individuals at their trimester 1. So we collected 3 to 6 samples through the longitudinal study period of time. And in total, we have over 2,000 samples of these 400 participants. And what we did is that we collect their clinical diagnosis and metadata, and we have their questionnaires, whether they take medications, what any special events happened during their pregnancy, and then we cultured all the samples for bacteria and fungi, and we can sequence them for whole genome at whole genome sequencing level. And we also did ITS and 16S sequencing to characterize the structure of the bacterial and fungal communities in the vagina of these individuals. And we also quantify the bacteria and fungi by qPCRs. So this is a very high dimensional data, and we finished these all the sample collection in August, and we just finished the ITS and 16S sequencing a month ago. So I'm going to show you some very preliminary data here to give you some ideas. First, some demographic things. So what are the fungi in these individuals, in these healthy women? Albicans is the most frequently identified species. This is aligned with other studies as well. And we also, because we had the questionnaires, whether they have any symptom or not. So we can see that about over 70, almost 80% of the individuals who have albicans colonized in their vagina will have these symptoms. And in terms of glabrata, which is the second most common Candida species in this cohort, only 50 of them feel some itchy or other symptoms of VVC symptoms. So albicans is cause more symptoms compared to glabrata or other fungal species. And here, we also detect whether we also check that the colonization, fungal colonization by culture. And here, one row represent one individual, one dot represent one sample. The green dot means the culture positive for albicans. The open dot means culture negative for albicans. And the drug means because the patient will have, the pregnant women have symptoms and they want to treat their VVC, so they use antifungal drugs. It's either, these are either topical azoles or nystatin. These are all topical drugs because oral fluconazole, which is the most common way to treat VVC in non-pregnant women, the oral fluconazole increase the risk of stillbirth. So oral fluconazole is not recommended in these individuals. So we can see that if a woman had a Candida albicans colonization at the trimester 1, it's very likely she will have a persistent albicans colonization throughout the pregnancy time. And this is despite of a usage of antifungal drugs. So the antifungal drugs, a lot of chance it will not be able to clear the Candida albicans colonization. And this is even worse in the glabrata. So glabrata has even better persistency. With the glabrata, there's only one lady that has a temporal glabrata colonization. The other of them all have persist glabrata colonization. And this is by culture. So how about by ITS sequencing? So we actually were able to also define the community state type of fungal community with 5 different types. The type FCST1, community type 1, is dominated by albicans and also by glabrata. And some of them have other species as parapsilosis or tropicalis. And we also find a few of them have domination with Malassezia or Saccharomyces. And again, most of them, the purple, light blue ones, these are no colonizations. So these basically summarize the community state types of the vagina fungal communities. Okay, so these are some really preliminary data. I'm sorry I could not provide more at this moment, but there are a lot of things we can think about. So for example, what does this vagina colonization affect the outcome of pregnancy? And will these persistently colonized fungi develop the resistance during the treatment of topical use of azole drugs or nystatin, the pollen drugs? And what are the signatures of bacterial flora with or without Candida colonization? So this, I mean, sorry, I cannot provide too much at this moment, but I think probably next year in the Candida conference or some other conference, or maybe you can get me back again to update with the new data there. Okay, so with this, I would like to thank my lab and my collaborations from MSK, from China, and from basically all over the world. Thank you very much. I would like to take questions, I guess. I guess there will be a lot of questions.
Yeah, wonderful. Thanks so much, Bing. That was really fantastic. And yes, we look forward to hearing the update in a little while. So yeah, we'll open this up for questions. So after this, we're going to have, there'll be like a more focused roundtable with students, but regardless, we encourage students to ask questions now and faculty and anyone else here. So we'll open it up. Oh, there's some. questions, thoughts? I can kick us off. I've got all sorts of questions. I guess when you're testing, when you're selecting your like culture positive versus culture negative, you said you sort of bias towards culture positive in your selection. I was wondering what the actual sort of prevalence of fungal culture positive pregnant people are, if you weren't sort of selecting for those specifically. Oh, been frozen.
Mm, I think she's froze. I don't know.
Yeah,
if
okay.
she's down.
Let's
Yeah.
give her a second. Maybe. Oh,
Pretty amazing thing.
yeah.
Full so
Oh, oh, what's happening? Okay, I think she's coming back in. I just sent her a message too. Okay. Oh, I was going to say welcome back. I think we can at least maybe hear you now. Bing, can you hear us? Give her one more second. Give you all lots of time to prepare your questions. Well, there's a question as to whether turning off our cameras will help her internet connection. What do we think?
worth a try. Do the experiment.
Okay. All right. Cameras off.
Take two.
Is she here?
Just readmitted her again.
Oh, okay, good.
Okay.
Oh, Awesome.
How
Okay, Bing,
about now?
we all went cameras off to try to help your internet, if that helps.
I use my cell phone now.
Okay, well, we can
Use my
definitely hear and
cell phone now. Okay, great. So
we
okay,
can,
questions.
yes, perfect. Okay. But anyways, if you're wondering why you're not seeing any of us, we're trying to preserve your bandwidth. Okay, so I thought there was a hand when we were last here. I didn't see to who it belonged. Does the hand want to reappear? Okay, Mahmoud.
Great. Thanks a lot for the presentation. I really appreciated the first and the second part of your talk, which was really clear. I have two small questions. The first related to the first part. When you are talking about this heteroresistance, does this mean that can this be due to the first of all the use of the antifungi first, but also to the low activity of the immune system in those patients because they are transplanted, so probably we're using immunosuppressive drugs to decrease the activity of the immune system there?
Okay, so I think that's a fantastic question. So first, I don't know whether the low immune system will affect the development of resistance. I think my mentor Tobias Ho is actually working on this in his lab now. But my guess would be no. So my impression is that because the isolates we got from, say, in China or from Europe, these are not all from the bone marrow transplant patients. So they are not necessarily to be isolated from individuals with immunocompromised conditions.
Okay.
So I think my gut feeling is that this is not direct. There's probably not a direct link. And in terms of the usage of antifungal drugs, I think my observation is that Memorial Sloan Kettering Cancer Center used microfungi as prophylactic drug since 2008. So it's been about 16 years there. Before I checked a lot of the medical record of the hospital, and before the year of 2009, there's no parapsilosis bloodstream infection in LOHCD patients in this hospital. No.
Okay.
Albicans, glabratas there, yes. But since they switched to microfungi as prophylaxis, they start to see more and more parapsilosis infection in the past decades. So I think that's a more like a group effect. So they use this not for only one patient. In each year, there are several hundred of patients undergoing the LOHCT, the bone marrow transplantation, who use these drugs for at least 2 to 3 weeks. And there are also leukemia patients who's also neutropenia, also use these antifungal drugs for prophylaxis, for some of them, for a long time. So this is like a selection of heteroresistant isolates by the high dose of antifungal prophylaxis. That's my guess from the observation. Yeah.
Thank you so much. That's great. My second small question for the second part. When we're talking about the appearance of that Candida infection, vaginal Candida infection in pregnancy, once again, in pregnancy, the immune system is decreased somehow.
Yeah.
So do you think there is a sort of a link between that way and the appearance of that pregnancy?
Again, we don't know. But during pregnancy, there's not only immune system, the immune system is different. The hormones also a big difference. And during pregnancy, there's a high estrogen in the vaginal tract, and that actually promotes the production of the sugars in the
Yes.
tract, and that's actually provide more nutrition. So that in the vagina of the pregnant women, there's a high chance for both bacteria and the fungi to colonize. Therefore, the pregnancy is a known risk factor for vaginal infection. And I would love to take a look at the immune system level, immune responses in these ladies as well. So we were lucky. We saved another swab for this patient, for these individuals, and then we probably can test some of the cytokine levels there, hopefully in the future when we clean up all the sequencing data.
Well, that's nice. That's nice. I think I would go more further with the beta defensins instead of cytokines, because it would be more specific. I think.
Great point. Yeah.
Thank
you very much for the suggestion. Thank you.
Awesome. I saw Lisa's hand first, but in the name of allowing trainees opportunities, first we'll start with Philippe maybe, and then over to Elisa.
Thank you, Rebecca. Thank you for the talk. It was really, really interesting. I had a question regarding the first part on heteroresistance. So you showed that there was a fraction of cells that were able to survive whatever the microfungin concentration. Did you also try it with different antifungal to see if it's a general kind of response to drugs, or it's very specific to microfungin?
We tried Caspol and anidulafungin for some of the isolates. This is not just for microfungin. Echinocandin drugs, I think, from our data, there are some disagreement. For example, one isolate may be heteroresistant to microfungin but susceptible to anidulafungin. There are very few cases of that, but in general, if a isolate is heteroresistant to one echinocandin, they are likely to be heteroresistant to the other two as well. Yeah, we didn't test the azos, but one observation is that for those patients who had Candida parapsilosis domination in their gut during microfungin prophylaxis, because a lot of them actually got switched to azo prophylaxis after 7 to 10 days. After they switched to azos, most of the parapsilosis domination were gone. And we start to actually see more glabrata domination there.
That's really interesting. Thank you.
You're welcome.
Over to Elisa. You were a student too. All right.
Yeah. So I had two questions, but I think the time is almost up. So I'll just ask the first one, which is related to the first part of the talk. The pop-in ANI grade that you showed, which was showing 99.999 and 99.9999 similarity. So my question was about how are you able to take out the sequencing bias or errors in order to be certain of the such high similarities?
Oh, yeah, this actually, I would like to have more investigation and discussion with collaborators and friends in this field. So this approach we use, this is not HAS or whatever. We only calculated the homozygous mutations. No, we did not calculate HAS here. So that's why there's a very high similarity there. And we were now actually trying to figure out how to deal with the HAS situations with this similarity, this genome similarity comparisons. And because this ANI analysis was developed from the bacterial genome, those studies, and they don't need to worry about heterozygosity or those parts things. And that's I mean, we are dealing with a diploid, And that's our blessing and our curse. So
Exactly. All right. Thank you very much.
Okay, awesome discussion, awesome talk, great questions. Now is the time where we turn this over to a little bit of a sort of student roundtable. So we allow the students unfettered access to you, Ben, without, I don't know, us pesky faculty hanging around. Assuming the bandwidth can handle it, I encourage everyone to attempt cameras on. If that fails, maybe we'll cut that out.
I think my phone seems more robust than my laptop.
It seems okay. And you did make it through the talk, which was the most important part, with no problems.
Thank God.
Yeah, exactly. So we encourage students to turn their cameras on, have a discussion, and we encourage questions both about your awesome work, but also your career trajectory, sort of everything and anything.
Sure.
So with that, I will say goodbye and have all the faculty log off. And thank you again. It was really fabulous talk. Excellent work. Can't wait to hear more.
Thank you again, Rebecca.
Absolutely. All right.
Thank you, Bing.
Bye.
Thing is that I've been letting people in and I'm not going to stay. So Bing, if I give you hopefully, or is there
maybe
a fellow that I can, I was gonna say, is there a fellow that can volunteer to be the meeting co-host that you can let in people if they?
I think Eileen is the host, right? Or like the appointed host of this roundtable. Yeah. So maybe make Eileen just in case Bing's internet goes down.
You make host. People from my lab are just leaving our like joint little lab group, and they'll be joining in a couple of minutes.
Cool.
Thanks so much, Bing.
Awesome.
That was interesting.
Awesome. Bye.
Thank you. Bye.
And thank you so much for that wonderful talk. It was really great, really clear, really interesting work. I guess, does anybody have any questions?
Thank you for tolerating my midnight scramble.
Thank you for being up at midnight to talk to us. Yeah, Maria.
Okay, I think I saw Rachel. Hi.
Hey, I have a question. It's related with your research, but in the vagina colonization, I saw that there were like the graph with the participants and the days. There were times where the woman had like the infection, the positive culture, then it disappears.
Colonization, yes.
Yeah, and then it goes back. So do you know like why does this happen? Like you had like the positive culture, then we're like two without positive culture, and then it comes back again. I don't know if it's related with the flora of the vagina itself that led us to come back, or if there's like a mechanism behind to see if why the candida comes back in this woman when they disappear. I don't know if I made myself understood.
I think I get it. So I think there are two possibilities at least. The first is that this is by culture. That figure is by culture. It's not by sequencing. So the culture could have limited resolution in detecting the colonized organism there, right? I mean, we were already trying our best to standardize our sample collection procedure. So we have actually 3 designated nurses. Still, there could be possible that the sample we got did not capture the viable organism from the vagina tract. That's one possibility. Second is that I actually didn't mention during my talk because we not only got the vagina swab, we also got paired up anal swab, the rectal swab. We found in 2 or 3, I think at least 2 participants, we found that they have anal swab culture positive but vagina negative. And then they have vagina positive afterwards. And they isolate from the anal swab, the early anal swab and the later vagina swab are identical almost by genome analysis. So they could acquire new organisms from the transmission of the other skin colonized or intestinal colonized fungi there as well.
Okay, thank you so much. Pretty interesting talk once again.
I'm actually curious to sort of build on that. I'm not super familiar with like the how exactly you do all of this sequencing and sort of are like detecting the colonization when you do the sequencing. But is like any detection of the organism at all considered to be colonization, or is there a way for them to have like a couple individual cells that are maybe like commensals in the microbiome, but not full colonization? Does that make sense?
So I think commensal has a layer of meaning of colonization. Because commensal, it has to be there, right? As a commensal, the commensal has to be live together at the niche of the human body. That is called commensal, and that is actually colonization, in my opinion.
Okay. Okay.
Yeah.
That makes sense. Thank you.
And in terms of the detection,
oh, sorry.
Yeah, in terms of the detection limit resolution, so I actually showed the 3 different levels of resolution. First is by the smear, the microscopy, that's the lowest. And by culture, it's actually better than microscopy. And by ITS or 16S sequencing is actually better than culture as well. And I think there's a study by Margot DeLevy, who is actually doing postdoc in Tobias Lab now. She had a study showing that even a lot of samples, even for it looks very low, but she looked a lot of the metagenomic sequencing data, and she can find a very low level of fungal 3s there. She felt like even for the sequencing method, sometimes maybe we just roll out a lot of the sequence during the QC and all the process, the pipeline process. And there could be actually still very, very low level of fungi stay there instead of we just could not detect them very well using the current pipeline, the sequence analysis.
Okay,
Okay,
that makes a lot of sense. Question. Thank you. Yeah, I think Kelsey has a question.
Yes. I'm asking a question for somebody else. So like I don't know this completely, but he was wondering if there's a correlation between hormone levels with Candida albicans colonization in pregnant women, and maybe is there something related with glycogen levels as well?
Wow, great question. I wish I can answer that now, but we don't have the data now for the hormone levels. But I think from the literature that I read, the hormone levels correlates with the nutrition level conditions in the vagina niche. So that could be a correlation. I think there could be a correlation with the hormone levels and the Candida colonization levels there.
Cool.
But
thank you.
There could also be there could be another that's pure from the hormone side. Another side is the bacterial community. So some bacterial community, some individuals may have the vagina bacterial community that have more has more ability of colonization resistance to the Candida. So the answer is sorry, we don't know yet.
Sounds good.
Awesome. Does anyone else have any questions? Yeah, Maria.
I have another question going to the hormonal. You're working with pregnant women, but what about, I don't know, women? We know that you showed the graph of the map that it's affecting like women around the world. What about women that are not pregnant? If there are like another factors, for example, instead of the hormones of pregnancy, for example, hormones from birth control, I don't know, can be. So I don't know if for the future you will be interested, also in another
I
sampling.
really like your questions.
Another
That's a great
sampling that
point.
will be like
And
this
and
one. Yeah.
yes, I think you made a very great point that yes, we only look at pregnant women now. What about women without pregnancy? How about those birth control pills? Well, the reason we chose pregnant women to study, there are two reasons. First, pregnancy is a risk factor for VVC, that we know. Second, it's actually pure by microbiome study, the philosophy of microbiome study. I am a believer of longitudinal study of microbiome because I think this is a very complicated community. I don't like cross-section study, cross-section data, because there are so many random factors there. And I think longitudinal data set would provide more accurate and more informations for the community we are looking for. It is very easy, much easier to track longitudinally compared to non-pregnant women. So we want to start from easier. And we want to show, okay, so this could work. And then use this to persuade more people to participate in our studies. Even for this cohort, so I show that we have 405 individual participant volunteers, that the effective volunteers data, the individual data. We actually recruited 600 pregnant women signed on the study at the beginning, and a third of them dropped during the study. I mean, think about it, these are these are the ladies that come back to the hospital almost every month, and they still could drop from the study. And then how about those non-pregnant ones? Like, those are not very easy to track with. Yeah, that's how I conceived this project at the beginning.
Yeah, that's true. It can be really hard to control, but also I think that you will have for the future very interesting results of also like how birth
Actually,
control can
from
affect.
your side, it's actually, hi, Maria. I think from your side, you're all in the universities, right?
Mm-hmm.
University students is another group of that is more easy to track. Yeah.
Again, thank you so much.
You're welcome.
I actually had another question going back to sort of the heteroresistance part. And I was wondering, first of all, if you know anything about what that gene with the partial deletion in the heteroresistant Candida species, like, do we know anything about what it actually does or anything about that gene at all?
Oh, that is a gene that has not been very well studied. If you check that on CGD, the Candida Genome Database, it shows that it may have some activity with some activity with the cell wall integrity. So like very hand-waving descriptions. So we hope we can, yeah, my postdoc is going to do more genome editing and trying to see whether those that gene has any direct relationship or causal relationship with this phenotype.
That's really cool. I'm also wondering if, say, we could get a test that's able to, detect for like the isolates that have that partial deletion in the clinic sort of thing. Is there, are there other options for those patients, like other antifungals that we can give them on top without interacting with those chemotherapeutic drugs? I know you said a lot of the azoles interact and there's not a lot of antifungal classes. Like, do we have options for those patients?
Well, we have 3 major classes of antifungal drugs. Azoles, echinocandins, polyenes. Anfol is probably too much for you cannot use anfol for prophylaxis purpose, right? And azoles, they targeted the P450 class of enzymes, so they could have all the azole drugs are supposed to have the drug-drug interactions with some of the chemos. Echinocandins, well, that's it basically, right? So in an ideal scenario, in an ideal world, so we still put this patient on microfungal prophylaxis, and we monitor their poops. We see whether there will be parapsilosis bloom there. Once we see that, we could switch the prophylaxis from microfungin to azoles, trying to clear up that.
Okay, that makes a lot of sense.
That will require a lot of effort from nurse, from the physicians, and that's why I said that's in the ideal world. The precision medicine is very costly.
Thank you so much. Does anyone have any other questions? Okay. Great. Well, if no one else has anything else to add, thank you so much.
You can always email if you have any questions in the future.
Thank you. And this was a wonderful talk. Again, thank you so much for staying up late to talk to us.
Yeah, it's my normal time my normal bedtime, like 1-ish.
Thank you so much.
You're welcome. Okay.
Yeah. Thank you. Have a great evening.
Bye. Have a good day.
Bye.